fifth case of food poisoning this week. Any idea where he got it from?
When dealing with foodborne outbreaks, it's helpful to learn as much about the pathogens that are responsible for them as possible. One way to understand a pathogen is through its genome. Similar to all living organisms, including humans, bacteria have a genome made up of DNA. The DNA for a bacterial pathogen is comprised of anywhere between 3 and 5 million nucleotides. The A's, G's, C's, and T's strung together like beads on a necklace. The order of the beads forms the DNA sequence that serves as an operating manual for that organism's physiological functions. Different segments of the sequence contain instruction sets for different functions. These individual segments are sometimes referred to as genes. In this example, a segment of the DNA sequence provides instructions for a pathogenic bacterial cell to make a protein. This particular protein is destined for the outside of the bacterial cell and will enable the pathogen to adhere to the human intestinal wall. Bacteria that have this gene are able to stay in the gut longer than those that don't, and thus are more likely to cause illness. Using a technology called whole genome sequencing, we can determine the DNA sequences of bacteria and other pathogens and identify the genes they have. This information is invaluable for identifying, differentiating, and understanding the risks associated with food-borne bacteria like Salmonella, E. coli, and Listeria. In fact, there is much we can learn from knowing the whole genome sequences of these bacteria. For instance, some genes enable bacteria to resist antimicrobial drugs, thus making them drug-resistant. Other genes make the bacteria more pathogenic and increase the probability of disease in the host, like the adhesion protein on the outside of the cell. Some genes confer increased virulence, which is correlated with worse illness or disease outcomes, while other genes allow bacteria to survive in and on specific food products. And finally, some DNA sequences are associated with particular geographic regions and provide information about where they came from. These are all types of information that we are extracting from whole genome sequences and applying to food safety.